Welcome to SARC Talk. SARC is a nonprofit pan-sarcoma research organization that develops and manages clinical trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review innovative scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode number 6. My name is Scott Okuno. I'm medical oncologist at Mayo Clinic and chief medical officer of SARC. All episodes are posted on our website, Spotify, YouTube, SARC channel, and wherever podcasts are available. Please place comments as we want to serve your needs. Today, our guest is Dr. Chen, Associate Professor of Internal Medicine in the Division of Medical Oncology and Biomedical Informatics from The Ohio State University, and he's Senior Vice President of Medical Informatics at Tempus Labs. Dr. Chen was a SARC Career Development Awardee in 2014. Dr. Chen's research interests are in precision oncology and bioinformatics. We look forward to learning more about academic sarcoma centers and how we can partner with industry. Dr. Chen, welcome.
Thanks so much, Scott.
I've been looking forward to visiting with you. You know, when we set up SARC Talk last year, I realized that there's a lot of wealth of individuals that have come through SARC, through the Career Development Award, and that continue to pursue sarcoma as their academic interest. Dr. Chen, tell us what you did at Ohio State prior to Tempus Labs.
So at Ohio State, I had a couple different hats. First and foremost, obviously, was patient care. I've been a sarcoma oncologist for over a decade now. I can't believe I'm saying that. It's kind of a little surreal to me. It feels like yesterday since I took over for a colleague right out of fellowship. The other hat I was wearing was also, I was the medical director for research and clinical informatics for the James Cancer Hospital. And that was a hat that actually was very fascinating to me, and it let me see into some of the decision-making. And there is clinical informatics, but there's also what we think of translational bioinformatics. This was very much a little bit more of a clinical informatics role, dealing with electronic health records, dealing with, for example, the telehealth COVID rollout, which was 2 years of very, very busy work, I think, for a lot of people. And the third hat, obviously, was to maintain a research lab. I really focused on developing biomarkers and getting interested in ways of characterizing kind of disparate sarcoma subtypes.
And how did you decide from that point to continue your academic pursuits and your interest and expertise at Tempus Labs?
I think that's a little bit of an easy question. Part of it was that what I was realizing is as a sarcoma researcher, as a clinical informatician, we were really hitting a wall in the things that we could do at academic medical centers. Although we do share data, we don't have the large corpus of data. Although we do have bioinformatics techniques for doing things, oftentimes we're understaffed and we're trying to grab a statistician or a bioinformatician to try to do work. So for some of these endeavors that we really want to push forward that we think are going to be groundbreaking or really change up the way we do medicine today, I believe that has to be done in high tech. I don't think there's another way around it. Obviously, there'll be some developments, but I think the larger developments, if you think like the Googles or the Amazons of the world, they have to come externally. We're not going to see that coming necessarily from an academic medical center. We just don't have the ability to kind of corral everybody together and do that, because research, obviously, for some people is full-time, but for a lot of the medical oncologists or sarcoma oncologists, a lot of that is a part-time passion. So it's hard to really compete with a well-funded healthcare machine that potentially can get the data together faster than you and analyze the data faster than you. That's a long way of saying, basically, that one of the things that I noticed was that all the data is being aggregated by pharmaceutical companies, by pathology companies. So if you really wanted to study rare diseases, as I still and very interested in, you really need to work with these companies. You really need to partner with them in order to continue that sort of research. And I think that was one of the largest attractions, was the ability to then play a role in kind of shepherding these rare diseases forward within a lot of these company infrastructures. Tempus Labs is an AI company focused on healthcare. And so when you think of it that way, artificial intelligence work, you're thinking of developing algorithms, you're thinking of developing better ways that physicians can interact with clinical data. You start thinking about, wow, when you think about the clinical data, well, is it any good? You start thinking about whether or not these rare disease types are represented. Is it representative of your experience? And so these are things that I felt that could be brought to bear in order to better study rare diseases or better drive some more precision oncology forward.
Having somebody like yourself who has expertise in 10 years of clinical expertise, knowing the nuances of subspecialty or subhistology sarcoma at a major corporation like Tempus Labs is going to be an asset to the sarcoma community. And we'll get down there a little bit further later in our questions. But you brought up that you were initially a clinician and still are a clinician at heart there. And the Ohio State University Sarcoma Program is a large multidisciplinary group. Can you share with our listeners here what is unique about the sarcoma program at the Ohio State University?
What's great about it is that it is truly multidisciplinary. At other institutions where I've been before, they've also been multidisciplinary, but not to the extent that Ohio State has a multidisciplinary team. Our team really is fairly extensive. There's a nutritionist on staff. They're usually met with my social worker, the nurse practitioner, our nurse who helps triage the calls. They meet everybody. So it's no less than 6 people, I would have to say, at these new patient visits that are really touching the patient, especially if they start chemotherapy or if they're going to start some treatment. There's additional folks that work with them, like our chemopharmacist that sits with us, as well as our medication assistance program folks. It really takes a huge team of people, and this is what we really talk about. Kind of that multidisciplinary within the clinic, but then you also talk about multidisciplinary within tumor board with radiation oncology, medical oncology, pathology. And I think that's what a lot of people do think about when they think multidisciplinary. But in my mind, too, that carries over into the clinic.
Yeah, that's a good point, Dr. Chen. I didn't think about that. These major academic centers like the Ohio State sarcoma program, your medical oncology sarcoma clinic is really multidisciplinary in bringing all the resources available for sarcoma patients and allowing them to get through diagnosis, treatment, and surveillance. And more importantly, but in addition, you have the wealth of resources of the surgeons, the radiation oncologists, the pathology, and imaging people there as well. And I do think part of what we do at SARC is to highlight centers that have both the medical oncology multidisciplinary approach, but also the, as you said, cross-discipline multidisciplinary approach. And that's a good way of saying it. So I appreciate that.
And one of the things I would also highlight is our tumor board, the sarcoma tumor board, is one of the best attended ones, even though it's at 6:30 in the morning. And it's run by our musculoskeletal radiologists. But there's always a person who does nuclear medicine. It's surgical oncology, medical oncology, orthopedic oncology, radiation oncology, plastic surgery. And so the list goes on and on, the people who are at the tumor board. And it's very collegial, which is very, very helpful. It's definitely a working tumor board in terms of we're trying to get through quite a few patients given the patient volume.
No, and I think at those centers and those, even if it's 6:30, it's still a good opportunity for us to get input of what the next direction should be with people that are outside your department. So that's great. You bring about the word distraction, and there are a lot of distractions in our life. And I know that you said that when you go to Tempus Labs externally, they're focused on one thing, where we in academics have multiple different pressures and priorities that are pushed upon us. But you did start your career, at least in sarcoma, you were awarded the co-recipient for the SARC CDA in 2014. And I look at the title here, Predicting Doxorubicin Sensitivity in D-Differentiated Liposarcoma. So can you tell us about how that award went and how it shaped your career? Because we're talking to other career development awardees, and most of them didn't stay in the field that they started with, and they pivoted, but it was very helpful to get them in the door, as they say. So Dr. Chen, share us your thoughts.
That SARC award is really, really, truly pivotal, I think, in the way I think about sarcoma data today. I think when I had received that award, I had developed an algorithm, a way of kind of trying to do prediction, and was really based on genetic alterations. So basically gene changes, very specific gene changes that were commonly used for things like gene chips, or which was, I think, very popular at the time in sequencing, which became more and more popular, looking at the DNA changes. What I didn't know is that liposarcoma, one of the cancers that I picked up, one of the key gene changes was something called an amplification. We have more copies of it. And it was really eye-opening. This was another axis of data, which is not commonly reported. And because we really focus on, I said, kind of like this expression values, which is RNA-based, and you really focus on DNA mutations. But this is yet a third axis. And so for me, it was this idea that, wow, okay, you can have an interesting question, interesting way of analyzing the data, but some of the fundamentals are, do we understand the tumor well enough to be able to characterize it in a multimodal way, such that we're taking advantage of all the sides, all the facets of the sarcoma data? I also learned very quickly that sarcomas really don't, some of these sarcomas don't really mirror what we see in other cancer types. And this was a perfect example where the dominant alteration was really an amplification rather than a mutation. It's a little bit wonky, but it's very different than what you think of when you think of a lung cancer or when you think of a breast cancer. So the SARC award helps me as I muddled through trying to analyze particular data, realized I had to get more data, realized I had to partner with companies that had that data that somehow had collected it. And that's indeed what I did, getting a large data set to at least find out what the cutoffs were, right, for some of these changes, because they really hadn't been published on. People noted that there were changes, but people didn't really know how big the change was. So that was one of the things that we did, was just try to understand, okay, are there natural cutoffs? Are there natural values that we should be looking for? Again, these amplifications, there's more copies than you expect. Are there very high tumors? Are there medium tumors? Are there low tumors? So that was kind of one of the first forays into this idea of, okay, how can we think about rare diseases? And realizing that they're all a little bit of an island unto themselves, but also trying to think about what connects them together. So I think the SARC award is really useful for allowing me to spend time in the lab and spend time doing that computational work to get a better appreciation of, again, this multimodal, multidimensional nature, which is sarcoma.
Yeah, Dr. Chen, you think about that as well, as sarcomas are unique in what you started to find out, that the typical things that you would typically think in other tumors might not be applicable. And you found out that maybe there are things that are unique for sarcomas that we need to pursue and build out a career. And it was like you said, the time to think, time to study it, and time to analyze and big data set was very helpful. And I can see how that's translated in your career path as well. And that goes back, as you and I met several times when we were at the Alliance meetings, and you were the co-chair of the Alliance Translational Bioinformatics Committee, and you also chaired the ASCO Cancer Link Oncology Task Force efforts. And this is not just sarcoma, but this is broader. And I'd like to hear some of your thoughts about how those efforts went. And then number two is, how does that help us in the sarcoma community?
Absolutely. So at Alliance, when I took the position, again, this is with collaborators where I work closely with. I don't want to, Ben Vinson, James Blashley, Dan Stover. These guys are in different tumor types, not necessarily sarcoma, but everybody has a passion for bioinformatics. One of the big gaps that we noticed was that even to study tumors, we needed the data. And I quickly realized that at Alliance, we actually don't have the data, all the data. So whenever there's a trial that's done, you're lucky if the data gets back to Alliance after, it might be locked up in some statistician's vaults, but some of the primary data is really with the principal investigator or the lab collaborator who had run some of the experiments. There was not a central repository for looking across all the different trials that we had done. There's certainly biobanks, but there wasn't a data bank per se. So I set out in the grant that we had put in, and this is something called ASTOR, Alliance Storage, basically, if you want to think of it that way, repository of molecular trial data and with matched clinical outcomes. Something as basic as, can we put all our trial information together and so we can do cool things with it downstream? I know it sounds super basic, but these are things that are not in place in a lot of places. And I really think, particularly for rare diseases, I go back to that because the only way we're going to understand rare diseases is if we can pull data more efficiently. So this was a little bit of a passion project for me, partly because with all the sarcoma trials that are ongoing, they're all accrue 25 people, which is the biggest trial, right? Or anything larger than that, you really have to start going across multiple tumor types. How are you gonna get this data if it's so siloed? Whereas a breast trial where you can have several hundred people at a time, right? It's a little bit apples and oranges. And so really the goal of ASTOR was to try to take advantage of what we've already done so we can learn from that. And it's surprisingly challenging, only to figure out kind of the governance structure, what sort of structure to put the data in. There's also historical data that don't fit modern molecular techniques. So how do you harmonize that? How do you think about the data? So that was kind of the big focus at Alliance.
I think you and I have to talk offline here because part of what we have at SARC as well is data. We have huge clinical database, but we also have tissue, we have specimens, and your comments are spot on that we might need a SARC store because the reality is, how do you mine that? Because as you said, some of our studies are multi-histology. So how do you pull out certain histologies and then combine them with other ones? And we're in the process of doing that. So we would love to follow up and make a decision of how we should proceed because you've had experience doing that through the Alliance. So that would be helpful. I know that you have done a lot in the bioinformatics and that's been your passion, but some of us have not grown up in that space. We're learning it. So we have to get up to speed pretty quickly here. And for those that are listening here, maybe you can give us an idea of where you see translational bioinformatics going in 2023, not only for us at SARC, but also NCI and other groups.
I think that's a great question. So I'll give a little bit of background. I first got into bioinformatics when I was in college, and I was lucky to have gotten to work at the Whitehead Institute, where they're working with the first Affymetrix chips. These are expression chips. Back then, I don't think I realized how lucky I was to have that opportunity. At that point in time, one of my cousins, who is a cancer researcher, said, "Hey, you really like biology. You like also computer science. There's a field called computational biology that's available to you." There was no such thing as truly bioinformatics, and certainly the idea of translational bioinformatics was even a thing. Truth be told, I've gotten grandfathered into the bioinformatics field. I always, laugh a little bit when I think about that because what does that really mean? And I used to be the course director for translational bioinformatics at Ohio State and taught that for several years. And so one of the key things, I mentioned this too, when we think about the direction of translational bioinformatics, which I always told my students is, it is such a huge field. There really depends on which aspect of the data you want to be a master of. So you can be a master of part of the data, or you can be masters of part of the algorithms, the transformations, or you could be users at the end trying to put these together. Let me give an example. There's people who really focus on developing better sequencing techniques to find interesting biomarkers that can be used for tumor characterization, for example. There are people who are very focused on trying to develop algorithms that can manipulate the data to help you find clinical results. There are also people who take the data that was generated, that was mined, to better link to clinical results at a deeper depth. And all of these I would call translational bioinformatics. They're just because in each of these cases, you're really trying to bring it into the clinic. And so there's always a translation portion there. So all of these are translational bioinformatics. As we move forward into the future, what I'm seeing is that a lot of the data aggregation portion is a little bit is becoming commoditized, just like I talked about here with the Alliance efforts, with Tempus efforts to develop a large multimodal data set for research use. This is something where I feel like we're finally getting some convergence in terms of standards, convergence in terms of how we think about the curation of these databases that need to be there. Just kind of a side note, one of the things also I did with on CancerLink Oncology Task Force, and before that it was the Health IT Task Force for for ASCO, and now the ASCO CancerLink endeavor was to work on. I helped chair with Doug Blaney the first MCODE meeting. Okay, and MCODE is this minimal clinical oncology data elements initiative. It sounds really nerdy, and a little bit is. But it's the idea of how do you transmit patients, right? What is the patient concept, and how do you share patients? What are the standards that one would do that? And that was one that MCODE, I'm actually proud to say that I got to name. It's kind of stuck, but now it's developed a little bit of a life on its own. I work on that today within different accelerator groups, trying to further MCODE in terms of what its use is. But this is going back to the point where we're developing standards for data transmission and for characterization of patients. So I think a lot of that will become, again, more set. A lot of the statistical techniques that we're using to analyze the data are also becoming commoditized. We're finally stabilizing in terms of how we think about some of the more established data sets. Obviously, neuromethylation data, that sort of stuff will require a little bit more work, but you'll see stability there. Where does this leave us? This really leaves us with this third bucket, which is how do we mine, how do we do a little bit better with clinical data? So progress notes with discrete data elements that are within the EHR. How do we combine those? And how do we combine things like CT scan images, digital pathology? All those are a wealth of information too. The field of radiomics, the field of digital pathology as mentioned. These are all kind of large data sets as well that really need to be connected to the clinical data, which need to be connected to molecular data. So that's certainly one thrust of where I think the field is going. That's something that that the company I work for is also working on right now. So we're very, very much aligned there. The other part is we have enough data now that we can start developing insights. You look at the ChatGPT movement here, and it's really super exciting that if you have a large enough corpus, the techniques in order to do some of this data mining through artificial intelligence means are available. So I really think that's the next thrust of this is to expand the data sets. And secondly, then use more sophisticated machine learning techniques that require large corpuses of data to help us mine health IT work. I think this was the hope of large efforts before that had failed. But I think if we think a little bit more modestly about it, what is feasible? What are the questions we want to ask? I think that we are there.
It sounds like we all should be bioinformatics in some level of our careers. And I think I'm going back to your MCODE, and I guess that's going to be your moniker going forward since you helped develop that name. The reality for us would be we needed to have the rules of what we wanted to collect. Now we have that. You're going to expand upon what you're going to collect for the minimal data set. But we as a sarcoma community have really not really started to do the real work that needs to be done wholeheartedly to translate into treatments and et cetera. I think right now we have the plans. we've built the railroad. Now we got to build the engine, and now we're eventually going to need some depots and, people to start getting on the bus, as we would say, the train, as we move forward with, this exciting field, because in reality, data is just data unless you start to use it for clinical use. And I don't know where do you see us in the next year or two? As you said, mining this data, using ChatGPT or other venues to help provide insight and eventual treatment options, or you think we're still a couple years away?
I do. Usually the challenge is not and again, it's like any good laboratory experiment. The challenge is not the statistics or running the experiment for the most part. At least a bioinformatics experiment, it's 90% data cleansing and 90% data preparation. And when we think about sort of yes, there's a biorepository, yes, there are clinical trials, but the trick is going to be, and I think we alluded to this earlier, annotating all the specimens, making sure that we can look at cross trials, we can find the right trials. And then if we have the data, how is that put together? And are we matching the same clinical data that's required with the particular specimens? And just like kind of my experience there, are we matching the right molecular data with the right specimens so that you can do the right analyses? One of the beautiful things, and where I tear my hair out with sometimes with sarcoma, is each of the different tumor types, or the ones that are fusion driven versus amplification driven versus as a single alteration, which is broken, are truly different beasts. And then if you once you start thinking about, okay, what does a structure have to handle? It's not a one size fits all. You can't ignore the fusions. You can't ignore the amplifications. So any data structure that you build has to make all these contingencies so that you can plug them in and then look across them. So I think a lot of this is going to be just prep work, and and I think sarcoma folks are the best because we have the best appreciation for the heterogeneity that one sees within the data.
I agree. And it's exciting because, as you said, all those three different types of things that you talk about, single alteration, copy number, or other aspects, is in every single histology. So that's going to be unique. One of the things SARC is known for is that we do have a collaborative partnership with industry. And we co-develop a lot of clinical trials through SARC, along with the help of Jonathan Fletcher, working on the basic science and industry support for that as well. And as we step back and look at SARC, look at the sarcoma community as a whole, industry, because we're such a rare histology, what is your perspective of how industry as a whole looks at the sarcoma community as one big group or small histology or a blend? And, part of your experience is that you've been around a long time and you've been interacting with a lot of industry people. So we'd like to hear your thoughts.
So that's a simple one. I think industry does really think of sarcoma as a single entity just because the rarity of it. However, with that being said, there is the growing appreciation for when we think of something like the trametinib trial for EHE, that there is recognition that there potentially are molecular pathways that are highly targetable with existing drugs. When we look at the desmoid field, for example, with some of the new inhibitors for desmoid tumors, really there are specific niches that people are finding within the sarcoma world where their drug can fit. So, but that requires large databases, again, or it requires lab work that really has highlighted and shed some light on what are these molecular pathways so you can move forward with that. If you think about the drug development these days, they're all targeted therapies. There are immunotherapies, there are gene-targeted therapies, and then you have your traditional chemotherapies. I think Walt Sadler, who was my research mentor at the University of Chicago, used to tell me that all cancer therapies are targeted. Some target many more things than others. And that was a way of thinking about chemotherapy. And I love that paradigm. But as we kind of shrink the targets smaller and smaller, obviously you get to the gene level. With large databases, you can mine now and say, okay, outside of your favorite large cancer, is there another tumor type that is similar to this cancer that would benefit from this drug? you can only do that with a large database, a large sarcoma database, in order to find these natural fits and what we call drug repurposing. I think that to kind of answer your primary question, this is where a lot of that field is going. And this is why we do need these large databases. I know I sound a little bit like a broken record, but if this is listening to this podcast and can remember anything at all, there's a great need to try to build these up. So the partner with industry, I think really is kind of twofold, is to provide that clinical validation, that what you just said there. But I think it's also to ask industry to provide some of these sarcoma data sets. We can find these pathways that might be of interest. Your typical folk who do these analyses might not appreciate the nuances and subtypes, right? If you're a bioinformatician, you're not going to appreciate that, okay, yes, it's a liposarcoma, but we're talking about a different subtype of liposarcoma. Are we talking about a different subtype of your favorite tumor, right? It's very, very nuanced. And so during my time, my lab has always partnered with industry, so we could analyze, for example, MDM2 levels in liposarcoma. Even though these are smaller sets of several hundred, it's the only way to get a large enough set to start saying anything about it. So I think the partnership with industry is kind of twofold. I think they need to help us, provide us with data support to a certain extent. And then I think the quid pro quo there is that if there's an interesting pathway, we can really provide clinical guidance on how to run the trials and how to reach out to these different populations.
It's not a broken record, Dr. Chen. I do like your theme, and I think it's important, and it builds upon what we do at SARC. Individual centers do see a lot, the major ones, see a lot of sarcomas, but even they are unable to do big data sets. So part of the value of industry is to pull all data sets from all places across the United States and the world. In addition, part of SARC's role is to pull investigators to pull data as well, and then partner with industry to help out as well. So I think it aligns nice, because we think that the world sets at your own academic center, but the reality is there's a lot of centers that we can pull and gain expertise, and that's the only way we pull data sets. And I know that in Europe they do a better job of being able to pull data sets. I assume that they've standardized what they want to collect, but I think opportunities to work with industry, as you said, along with collaboration with major cancer centers, is going to be the key part moving forward. That brings us to another thought, is I know that you are been in the field for some period of time. There might be some individuals listening that are young investigators that are starting their career in sarcoma. They might be a solo practitioner in a major center, or a partner in a multidisciplinary center like the Ohio State. What recommendations would you have for them as they start their career path?
I think the most important thing are to start getting involved in organizations like SARC, not so much necessarily to be part of the administration or to do research, but to meet people who are like-minded. A lot of the sarcoma knowledge, I think this is going to sound crazy to some of the listeners, is really almost like storytelling. It's passed on from clinician to clinician. Yes, a lot of it's written down, but some of the practice patterns, if you're a solo practitioner, you're going to find that depending on who you talk to, some of the conditioning regimens that we use, the supportive regimens, some of the medications may be a little bit different in terms of what's first line, second line, because there's not always clarity in what that looks like. So it's a very much an art. There is a huge scientific portion, but a lot of it is still very much an art. And the only way, if you are kind of a solo practitioner or you're working with one colleague, especially for the newer trainees, is I think you need to be able to reach out to people and say, hey, what's worked for you? What do you think of this case? And I think that human aspect, I think, will get you a long way.
A shameless plug, SARC is proceeding with what we are doing is a multidisciplinary educational tumor board. Just to have that capacity is to have groups of individuals to share and hear how other disciplines, how other centers approach treatment of cancer. Now just broaden our perspective and to have, as you said, Dr. Chen, this dialogue, because part of it is science, part of it is art. But we always want to say is I don't want to be doing what I'm doing 20 years in the future, what I've done today, because I'm hoping that I'm going to learn from all this stuff that we are doing.
And I do think that as we do better of codifying how each of the tumors are a little bit different and start developing more drugs for each of these tumor types, you can finally have this idea of first and second line. If we know nothing about the tumor type or very little, and we're using very generalized chemotherapies for them rather than targeted therapies, it's very hard to then say this is definitively first line, this is definitively second line, or these are additional options. I go back to the fact that as the corpus of data gets larger, we will be able to, and to your point, I really hope that in 10 years for each of the subtypes, we will have first and second line. It will be less confusing for the solo practitioners because we'll know a little bit better.
I like your word corpus of data, and I love the idea of pulling data together as long as we all agree what data we're pulling together. And you're right, we'll learn from each other. So as we end here, part of it is what lessons have you learned in your career path that you want to share with other people? Not just career advice, but just to say is that, you didn't start out where you are today thinking this is where you would be, or maybe you did. But what other thoughts or lessons that you've learned in your career path that you think would be helpful for our listeners?
Again, these sound very cliche, but these are very true. I think number one is to listen to your patient and observe very carefully. A lot of the nuances in terms of the analysis really rely on the clinician's ability to see these differences. Why do these patients do better? What are some of the clinical characteristics that could really drive the molecular characteristics that could be singled out? So I think for new physicians who are interested in the translational fields, part of it is putting on your clinical hat and being a good observer of these differences. The second thing is, I would say for translational practitioners or translational clinicians is please collaborate. There is no way that I think a solo practitioner, a solo researcher is going to be able to take everything from the lab to the clinic. It's going to be a team effort. You may be an amazing clinician and you're able to really pick out the clinical features just like I talked about, but you might not be the same person who can run the algorithms. You might not be the same person who is the master of the biobank or doing the pathology reads. It is really a whole ecosystem, just like we talked about for sarcoma tumor board, just like we talked about for clinic work. Research in of itself has to be multidisciplinary. There is a path. It's a team effort. People say that, but this is never more true than in sarcoma.
I like your word ecosystem. And it reminds me today as I go into the clinic that every single patient I need to learn something from. And I think that's what you're saying too. And then more importantly, is that I cannot know everything. I need to know other people who know the things and build out my expertise. And then in the end, it helps my patients, it helps me as a professional, and it also helps the community. So I do appreciate those thoughts and insights. Any final thoughts, Dr. Chen, as we end our session here?
I truly hope that more people go into the field of sarcoma or at least do a rotation through sarcoma. This is such a fascinating field, no bias here, obviously. But I think it's such a fascinating field, and I think it forces folks to think a little bit more broadly and think that, it's not really a one size fits all for most tumor types. But if you can think like a sarcoma oncologist, then when you go to other cancer types that seem a little bit homogenous, you might say, okay, what are the underlying subtypes that are there? My hope is really that people appreciate sarcoma for what it is. It's kind of a microcosm of the larger tumor environment. And if you can understand that, I think translating that into the larger tumor environment makes that thought much more simple. And that's certainly something that I've carried over from decade to decade into industry in terms of how I think about even larger data sets.
You're right, because we have the opportunity to be educators. And if everybody understands sarcoma, that understand lung cancer, breast cancer, brain cancer, bladder cancer, testicle cancer, as you might imagine, a whole lot better. So if you're listening and you are an educator in the field, enjoy those learners as they come through the clinic and help them share your passion for sarcoma. So Dr. Chen, thank you for spending time here on SARC Talk. And we appreciate your continued interest in sarcoma, both at The Ohio State as well as Tempus Labs. And I know our paths will continue to cross. And we appreciate the time that you shared with us.
Thanks so much, Dr. Kuno. This has been a great experience, hopefully to get more people interested in the field. And I'm always happy to serve as a resource. Just like I said, you can't do it alone. So feel free to reach out.
No, I appreciate that. For those that are listening, please share SARC Talk with those interested in sarcoma. Please leave comments and suggestions on our SARC Talk podcast, and we will address that at our next SARC Talk. You guys have a great day. Thanks.
Thank you for listening. Be sure to subscribe so you never miss a SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or ask questions, please email us at sarctalk@sarctrials.org. And remember, together we can find a cure for all forms of sarcoma.